Next presentation is by Jung from the Department of Agricultural Biotechnology from Seoul National University in Korea, with a very long title, Metabolic Changes in Spent Medium with Pig Muscle Stem Cells at Varying Passage Numbers for Cultured Meat Production. The floor is yours.
Yeah, thank you. Can I use this pointer?
Yeah, I think so, yeah.
Hello, everyone. I'm Jung from Seoul National University in South Korea. Thank you for giving me this opportunity to present in ISCCM 2023. What I'm going to talk about today is...
You may need to talk in the microphone, otherwise the online people will not hear you very well.
What I'm going to talk about today is metabolic changes in spent media with pig muscle stem cells at varying passage number for culture meat production.
Do you prefer to use this one?
Oh, thank you. Culture meat has emerged as a meat alternative due to food crises such as a rapidly increasing global meat consumption and the negative environmental impact caused by traditional meat production. In general, culture meat is produced by using the muscle satellite cells. To produce culture meat, the muscle satellite cell must exhibit the desirable characteristic such as the high proliferation rate and the ability to differentiate into the muscle tissue. In particular, the higher proliferation rate of the cells, the more muscle tissue can be produced, which is related to the efficiency and scalability of the culture meat production. Ultimately, the improving efficiency and scalability of culture meat depends on the selection of the cells in an optimal state. One of the challenges in selecting optimal state cell is to assess metabolic status, which reflects the functionality and health of the cells. So, how can we assess the cellular metabolic status? Metabolomics is a discipline that scientifically studies the product of the metabolism and chemical reaction in organism. This approach not only gain insight into the cellular metabolism, but also can offer us unique advantage in unraveling the complex biochemical process occurring within cells for the culture meat. In this study, we conduct the metabolomics approach for the spent media to deduce the metabolic changes. This approach is highly advantageous in determining the specific media component utilized by cells, and in understanding the accumulation pattern of waste product that potentially inhibits cell growth. Beyond that, it can allow the indirect evaluation of cell condition without destroying cells during culture meat production process. We all know that the stem needs such as the proliferation decreases with the increasing passage numbers. So herein, we investigated the metabolic changes in spent media by pig muscle stem cells, namely PSCs, at different passage numbers to indirectly assess the primary cell state. The PSCs were obtained from the biceps femoris muscle of the 7 days of the crossbred pig and sorted for CD29 positive cells using MAX. On the Thursday of the culture, when the PSCs reached at approximately 80 to 90% confluence, the PSCs were subcultures up to passage 5, and spent media was collected in each passage. The following gross media was used in cell culture. The fresh media was considered as a control group, and spent media and PSCs from passage 1 to 5 were considered as treatment group, and namely as follows. PSCs from passage 1 to 5 were named as follows, and they were used for the proliferation rate analysis, and FM and spent media from passage 1 to 5 were used for the pH analysis. Only PSCs and SM from passage 1 to 3 which showed significant changes in proliferation rate were used for the qPCR and metabolomic analysis. The statistical analysis was performed as follows. In metabolic analysis, the following criteria was set to select the differentially abundant metabolites, namely DAMs. Result and discussion. Prior to confirming the metabolic changes of the PSCs, we assessed how the proliferation rate was changed with the varying passage numbers. As you can see this figure, the proliferation rate was highest in PSCs2 and significantly decreased after passage 3. Such decrease indicates the alteration in cell metabolism resulting from the loss of the stemness of PSCs. Changes in media pH provide insights to the cellular metabolic activity and the types of the metabolite produced. As a result, the pH value should have significantly increased as it advanced from passage 1 to 3, approaching the higher passage. These results indicates the metabolic profile changed as the metabolism of the PSC changed through the subculture. Based on our findings, the PSCs from 1 to 3 were used for the qPCR and the metabolomic analysis. In case of the qPCR, there's the expression level of the marker genes in PSCs were lowest compared to the PSCs with the other passages. Based on our proliferation rate and the qPCR result, we can know that the PSCs from passage 3 to 5 exhibits the suboptimal state. So therefore, to gain a clue for the detection of the suboptimal state, we conducted the metabolomic analysis for the spent media. As a result, total of the 91 metabolites were identified using LC-MS analysis. Based on these 91 metabolites, the multivariate analysis was performed to illustrate the pattern of the metabolic change and to visualize data. As you can see, the left graphs, SM1 and SM2 were clearly distinguished from the FM and SM3 in PCA plot. Meanwhile, PLSDA showed not only distinct separation between all SM and FM, but also demonstrated the clear differentiation among the 3 SM groups. To summarize, that the metabolite profile of the cell culture changed dramatically while PSCs were undergoing cultivation, with SM3 in particular showing the less changes compared to SM1 and SM2. In our PSCA and PLSDA, the results verified that PSC underwent characteristic metabolic chain alteration during proliferation depending on their state. Therefore, to ensure the effectiveness of the indicator used to determine the condition of the PSC, we screened the DAMs. The VIP scores was calculated for assessing the variables by scores. As you can see, right figure, the 11 metabolites were identified with a VIP score higher than 1.5. Filtered based on the p-value and the forward change, there is a total 39 metabolites were visualized using a Venn diagram, right figure. Ultimately, these 7 DAMs potentially related to the condition of the PSCs were detected based on the VIP score and the Venn diagram. Taken together, it can be concluded that the 7 selected compounds will contribute determining the changes in proliferation ability according to the passage number of PSCs. Based on the 7 key metabolites, the overview of metabolic changes was generated. As you can see this figure, our 7 key metabolites are their interconnected metabolites were involved in metabolic pathway for energy production centered on the TCA cycle and glycolysis. Most of these metabolites were shown to have significantly higher abundance in SM3 compared to SM1 and SM2, indicating that PSC3 underwent characteristic metabolic alteration. In summary, the relative of our study confirmed that the relative abundance of the 7 key metabolites were differed according to the proliferation ability regulated based on the metabolic alteration of the PSCs. In conclusion, our finding contribute to securing primal cells with optimal metabolic capacity for proliferation, which is the most basic step in culture meat production systems. And we believe that our study approach could be applied to the actual culture meat product industry, and it can serve as a quality control to check the state of cells by detecting key markers in standard related to the cell proliferation rate. Yeah, that's all I have to prepare for you today. If there are any questions, I shall do my best to answer them. Thank you.
Thank you very much. Are there any questions? Really? In the back. Right. Oh, I think now the transmitter.
Thank you. Thank you for your talk. From the metabolomics data, did you garner any insights for potentially good ways to raise proliferation rates, for example, based on the media composition that you can change to influence this like metabolite outcome?
Yeah, thank you for your asking. I don't quite understand. Are you asking about the future plan or our research?
Or just does this data like give you ideas for how to like increase the rate, maybe in the bioreactor and stuff?
That's a good point. I agree your point, and we can catch the key signaling pathway for the how to we enhancing the proliferation rate of cells, but that's also our future, including our future plan. But this study aimed for give you to the approach for how the metabolomic analysis in spent media could be applied the culture meat culture meat industry. So I think our approach can be applied to select the key metabolites and how the cell state and it has the good condition or it has the suboptimal condition, something like that. And as you can say, it can be also include our future plan. Thank you.
Any other questions? In the back there, fortunately quite close.
Yeah, luckily. Thanks for your presentation. So the thing with metabolomics on spent media is that the cells can only produce a metabolite based on what they're fed. So have you looked at other media formulations and considered the impact that of what you are giving the cells in the first place has on the results?
Yeah, that's a very, very key point, and thank you for your asking. Yeah. Based on our research of this study, metabolism, which is crucial for the cell proliferation, is mainly involved in energy production, as I said earlier. Yeah. Since it is well known, the energy source such as glucose and amino acids are essential for cell growth. It is packed. The similar result will be occurred if we apply the other base media. But however, key metabolites may appear differently depending on the type of the culture media. So we aim to suggest that our research approach may be applicable to the just culture meat industry. So yeah, maybe differed with the culture based media. Thank you.
Okay, thanks.
All right. Thank you very much.